Today we are going to see the sense of smell. So the sense of smell is poorly developed in human beings, but when it is compared with the smells in many lower animals, because lower animals have most ability to smell. So what is leading us to smell anything is because of the olfactory membrane. This olfactory membrane, here you can see, it's actually present in our, for example, you can think this is our nose, nasal septum. These are the nasal holes, nostrils. So through this, in the superior membrane of this nostrils, these olfactory membranes are present. This is the structure of the olfactory membrane. Now we are going to see. So the superior part of each nostril has medially the olfactory membrane folds downward. So medially means this side. So medially it folds downwards along the surface of the superior septum, and laterally it folds over the superior terminate. Laterally it flows over the superior terminate. So it's in the region of somewhere like this here. And even other folds superior, and even over a small portion of the upper surface of the middle turbinate. In each nostril, the olfactory membrane has surface area of 2.4. So it is present in the both nostrils. Its surface area of 2.4 centimeters. So olfactory cells are the receptor cells for the smell sensation. Of course, these are the receptor cells. They are bipolar nerves. Here you can see these are the bipolar nerves. Derived originally from the central nervous system. So there are about 100 million of these cells of olfactory membrane interspaced among the subtenticular cells. So what is this? These are the supporting cells. Here you can see these are the subtenticular cells, and these are the olfactory cells. So by the support of these cells, they are present here. This is a mucus layer, and this is the olfactory membrane, which is present on the superior part of a nostril. And the mucosal end of the olfactory cell forms a knob from which the olfactory hairs, that is the olfactory cilia, are formed. It is 0.3 mm in diameter and 200 mm in length. So from here the olfactory cilia are formed like this. So in our nose, if you see, there are olfactory cilia which are on the superior side of the membrane, so which are that able to sense the smell. So protect the mucus that coats in the inner surface of nasal cavity, and also that forms a dense mat in the mucus, and the cilia react to the odors in the air and stimulate the olfactory cells. So this pathway continues like this and finally reaches the brain. So what is that pathway? We are going to see. So spaced among the olfactory cells in olfactory membrane are very small Bowman's glands that secrete mucus onto the surface of olfactory membrane. So here you can see these are the Bowman's glands which secrete the mucus. So mucus comes from the Bowman's glands, which are present in between the olfactory cells in the olfactory membrane. And next, coming to the stimulation of olfactory cells. How these olfactory cells are actually stimulated? This is because of G protein, because this cilia have a 7-pass membrane receptors. Here you can see. So whenever any odorant, that is any smell, stimulates this, it stimulates the 7-pass receptor, and it is binding with the alpha, gamma, beta components. The alpha component activates the adenylyl cycling, converting the ATP into CAMP, in turn allowing the sodium inside to the cell. So this leads to the depolarization activation of the receptors, hence which we are able to smell the things. So that reaches via olfactory nerve into the brain. So that is the thing which we will get here. And next, finally CAMP activated. So all those things happen. So what are the mechanisms of olfactory nerves that greatly multiplicates the excitatory effect of even a weakest odorant? So sometimes what happens, if you remember, when in the air there is very small weakened organ, then also you can smell it. Why happens that? So let us see. First one, because of the activation of receptor protein, odorant substance activates G protein, which in turn activates the multiple molecules of adenine cyclase of olfactory cell membrane, causes formation of many times more molecules of CAMP, and finally the CAMP opens still many times more sodium ions. So therefore a minute concentration can also form a specific odorant, initiating the cascade effect and extremely large number of sodium ions. This process of a sequence sensory and olfactory neurons to the amount of odorants. So this is the system by which we are even able to smell the very small odorant which is present in the nature. And next, coming to several physical factors affect the degree of stimulation. So what are those? First, Only the volatile substances can be sniffed. Sniffed means like we see smell from nose. So only volatile means only those substances and can be smelled. And also the stimulatory substances must be least somewhat water soluble and also lipid soluble, because our lipid membrane is bilayer, so they need to pass it, so that then only this cascade can start. So that is what they are saying. And if they are non-lipid soluble, it is somewhat difficult because they can't enter into it. And the membrane potential and action potential in the olfactory cells. Actually, the potential inside the unstimulated, that is normal cell, is minus 55. But when the cells generate the continuous action potential very slowly, varying from once every 20 seconds up to 2 or 3 per second. So most odorants cause the depolarization, leading to the activation of negative potential in the cell form to normal of minus 55 to minus 30 or less. So sodium are positive ions. First it is minus 55. As the positive ions are coming more inside the cell, so from minus 55 it goes up to minus 35. That is, it is getting more positive because of more positive ions inside the cell. This is nothing but leading to depolarization. So changing in the voltage towards a positive direction. So the action potentials increase up to 20 to 30 seconds, which is a high rate for the minute olfactory nerve fibers. So the rate of olfactory nerve impulses change approximately in proportion to the logarithm of stimulus strength. This demonstrates the olfactory receptors obey the principles of transduction, similar those of other sensory receptors. So the rapid adaptation of olfactory sensation. What is this? Let us see. The olfactory receptors adapt about 50% of the first second or so stimulation. So what they are saying is that the olfactory adapt to almost 50% of the first, second or after stimulations. That is maybe first or second, because first time when it comes it is somewhat not adaptable, but second, third time you can also remember it that that was that smell like that. So therefore they adapt very little and very slowly. Yet all known from our own experience that smell sensation adapt almost to extinction within a minute. So if you remember, only for some time you can means only you can smell it for some time, later again it vanishes. So after entering a strong odorous atmosphere. So because this is a psychological adaptations is far greater than the degree of adaptations of the receptors. So it is almost certain that most of the additional adaptations occur within the central nervous system, which seems to be true for the adaptation of taste sensations as well. So even this is true for the taste. In taste also there is adaptation. So first in taste, what happens when you eat anything, at first time it like so strong, but later it becomes diffuses. That is its intensity decreases. Same in the case of smell too. First time when you remember, first time when you see any kind of smell, it is like of strong thing. Later slowly it gets decreases. So the large number of centrifugal nerve fibers pass from the olfactory regions of the brain backward along the olfactory tract and terminate on a special inhibitory cells of olfactory bulb, that is the granule cells. So what are those we are going to see next? So on the onset of olfactory stimulus, the central nervous system quickly develops a strong feedback inhibition to suppress the relay of smell signals through the olfactory bulb. So search for primary sensations. Actually there are many kinds of smells, mainly 100 types of smells, but only here 7 are given, but these are actually not accepted by everyone. They are like camphoraceous, musky, floral, pepperminty, ethereal, pungent and putrid. Not only this, there are of course many smells known, but only these are the one which are given, but they are not accepted too. At least there are 100 primary sensation of smells, marked contrast to 3 primary sensations or 5 primary sensations of taste. Actually there are 1000 different types of odorant receptors, and also a smell is that people have been found who have odor blindness for single substance. Actually we have found odor blindness. Odor blindness means a person who cannot smell anything. So even for that person we have tested that person can't smell only one type of smell. So like that we have found many patients of odor blindness with many substances. That means only this, not only 7 are present, but many substances are present which are present in our nature to smell. So this discrete, so you can see single substance, such discrete odor blindness has been identified for more than at least 50 different substances. So it means even that odor blindness also indicates that there are 50 different substances, not only those 7. So this presumes that odor blindness for each substance represents the lack of appropriate receptor in the olfactory cells for that particular. So every particular substance in our in our nature has a particular receptor for us, so that only we can smell that particular smell. So the effective nature of the smell even more so than taste, has effective quality of either pleasantness or unpleasantness. When it is pleasant you will be there, unpleasant you will run away. So probably even more important than taste for the selection of food, of course. So because if it is good only you'll eat it, if it's spoiled kind of smell we'll never go through there. So who has previously eaten the food disagreed with him with the nauseating smell of the same food on the second act. So perfume of right quality can be powerful stimulant of human emotions too. Like in animals, odors are primary excitants of the sexual drive. And next, coming to the threshold for the smell. So the minute quality of stimulating agent, the air that can elicit a small sensation that is smelled when only 25th trillionth of a gram is present in each millimeter. Here we can take the example of methyl mercaptan. This is a substance in that very low concentration, that is only 25th trillionth of it, can also be smelled by us when it is mixed with the natural gas to give an odor. So this is detected when even small amounts of the gas leak from a pipeline. So this you can take for example of a threshold for smell. And the transmission of smell signals into central nervous system. So how it reaches to our brain, now let us know. The olfactory portions of the brain, we know they are present in our upper part of our nostrils. Among the first brain substance developed in the primitive animals, and much of the brain developed around olfactory beginnings. So actually this transmission, let us say, olfactory nerves fibers leading backward from the bulb are the cranial nerve 1, that is the olfactory tract. In reality, both the tract and the bulb are an anterior outgrowth of the brain tissue from the base of the brain. The olfactory bulb lies over the cribriform plate, separating the brain cavity from the upper reaches the nasal cavity. So the cribriform plate has a multiple small perforations through which an equal number of small nerves pass upward from the olfactory membrane. So actually what happens, first from the olfactory membrane, all these olfactory nerves passes through the cribriform plate of the ethmoid bone. After passing this, they reach into the olfactory bulb. This is the olfactory bulb. In the olfactory bulb, you can see they pass through the glomerulus, and next there is a mitral cell, and finally enter into the olfactory tract. So from this it enters into our brain of olfactory region of the brain. And you can see nasal cavity to enter the olfactory bulb in the cranial cavity. So the short actions terminate in the multiple globular structures of olfactory bulb called the glomeruli, if we have seen it. So the bulb has several thousands of such glomeruli, which terminus for about 25,000 of access from the olfactory cells. So also in the terminus for dendrites, about 25 large mitral cells and 60 smaller tufted cells. So the mitral cells are 25 large mitral cells and superior to the glomerulus. So these dendrites receive the synapse from the olfactory cell neurons, and the mitral and the tufted cells send axons to the olfactory tract to transmit the olfactory to a higher levels of the central nervous system. So by this we can identify the smell. So some research has suggested that the different glomeruli respond to different odors. So that was we said before, different receptors for different smell. So it is possible that specific glomeruli are real clue for the analysis of different odor signals transmitted, of course, because a specific receptor is present for the specific odor. And the primitive, and actually there are primitive and newer olfactory pathways. Primitive means an old pathway, less older means it is in the middle, and the most newer one is a newer pathway. Now let us see about these pathways. Actually the brain at the anterior junction between the mesencephalon and the cerebrum, there the tract divides into the pathways. So one passing into the medial olfactory area. For example, this is a brain, and one is passing towards a medial side, and one is passing to the lateral side. So this is taking at the place between the cerebrum and the mesencephalon. So and the other passing laterally into the lateral olfactory. So the medial olfactory area represents a very primitive olfactory system, whereas the lateral is divided into less, that is medium kind, and a newer system. So primitive, the old one is moving towards the medial side, and the less older, that is the medium, and the newer one are moving to the lateral side. So what is the older one is going, we'll see. The medial olfactory area of a group of nucleus located in the mid-basal portion of the brain immediately prior to the hypothalamus. So most of the corn species are the septal nuclei. So they go to the septal nuclei. below the corpus callosum they are present, which are the midline nuclei that feed into the hypothalamus and other primitive portions of the brain's limbic system. Limbic system is associated with the emotions, so that is the brain area most concerned with the basic behavior. So the importance of the medial olfactory area is best understood by considering that happens in animals. When the lateral olfactory areas on both sides of the brain are removed, only the medial system remains, and the removal of these areas hardly affects the more primitive response to olfaction, such as licking of lips, salivation, and other feeding response caused by the smell of food or by basic emotional drives associated with smell. So conversely, removal of the lateral areas abolishes the more common complicated olfactory condition. So what happens whenever we have did experiments where we have removed the olfactory areas? So when we remove the olfactory areas, we lose this sensation of whenever food is there, licking of our lips, salivation, all these are lost. So that means indicating the olfactory areas are important for this kind of responses. And next, less old, that is the medium kind. The lateral, it moves, this medium kind and the new time are moving towards the lateral olfactory area. So they're mainly going to the pre-pyriform and the pyriform cortex, and also the cortical portion of amygdaloid nuclei. This is important. So these areas where the signals pathways pass into almost all parts of the limbic system, again to the emotional system, especially into the less primitive portions such as hippocampus, and seem to be most important for learning to like or dislike certain foods dependent. So this is what which we say we like some kind of smell, we don't like. So that is what which is taking due to this lateral olfactory area of less old olfactory system. So for instance, it is believed that the lateral olfactory is mainly connection with the limbic behavior cause a person to develop an absolute aversion of food that have caused during nausea, vomiting. So all these are affected with this. An important feature of lateral olfactory area is that many signal pathways from the area feed directly into older part of cerebral cortex called the paleocortex. This is important. So many of these pathways are going directly into the paleocortex in an anterior medial part of the temporal lobe. Since this area is the only area of entire cerebral cortex where sensory signals pass directly to the cortex without passing first to the thalamus. Actually everything first passes, from here everything first actually passes through the thalamus and next enters into the sensory area. But this what this old olfactory system is doing, it is directly going into the sensory area without passing through the thalamus. So this is the one which is going directly. And next the newer pathway also goes lateral side, that is olfactory pathway, which also passes through the thalamus, passing to the dorsomedial thalamic nucleus and then passing to the lateral posterior quadrant of orbitofrontal cortex. So on the basis of studies on monkeys, the newer system probably helps in the conscious analysis of order. So this is the newer pathway for the conscious analysis of the order. So summary, what we have learned from this is it appears to be the primitive olfactory system that serves the basic olfactory reflexes. A less old system provides the automatic but partially learned control of intake, that nausea, that like dislike, that is by the olfactory receptors of less old system, that is medium type, and the aversion to toxic and unhealthy foods and a newer system, that is unhealthy foods up to this is because of this medium type less older type system. And the newer system is comparable to most of the other cortical sensory systems and is used for the conscious perception. This is for the conscious perception of smell and also analysis of olfaction. So let us see the final part, that is disorders. So what may be the disorders? Either those that involves a loss of ability to smell or linked to a distortion. So complete loss of ability, that is anosmia. A means completely loss, and reduced ability. Reduced means hypo, so hyposmia, and disordered means dysosmia, DD, or also known as parosmia. So particularly when unpleasant perception of an odor is present, the disorders of smell and taste go hand in hand in perception of a patient. So this is not something because both taste and smell are involved in the perception of flavor. Of course, flavor, we want to eat anything, we of course see both taste and smell. So this is all about smell physiology.